I remember one of the first times I met Jen, she pulled her laptop out of her bag and it was dripping with water, just like it was normal. We were like at a colloquium and I'm like sitting behind her and it's just like, it's like dripping water. I'm like, does anyone else see that? That's not normal.
Was the colloquium in a cave? Because otherwise that's not great.
No, I was running late and I threw my water bottle in the bag and it wasn't sealed all the way. And so it was a waterproof bag.
Yeah, but that just means it keeps the water in. If you put water inside a waterproof bag.
Yeah.
Amazing.
I never broke a computer that way again.
From ScienceSortOf.com, you're listening to Science Sort of.
To Science Sort of, you are listening to episode 336. Our theme this episode is sweet sake and spoiled spores. I am your host, Ryan Haupt, and joining me to talk about things that are science, things that are sort of science, and things that wish they were science is former guest of the show, returning champion, Jen Gallagher. How you doing, Jen?
I'm doing well. Thank you very much.
And we are joined by two of your students. I'm super excited to talk to them. We are joined by Matt Winas. Did I get that right?
Nope.
Ish.
It's wine ands.
No.
Ands. Wine ands.
We are joined by Matt Winas. Matt, I apologize for getting your name wrong. We are also joined by your former lab mate, Apoorva Ravishankar. Better-ish? Close?
Apoorva.
Oh, guys, it's terrible.
I just call her Apoorva, and it's
I tried so hard. I spent so much time practicing and preparing, and the distractions of my dog playing with the squeaky toy in the other room completely flustered me. I apologize. Super excited to have you both with us. Apologies for getting your names wrong, but, you know, I got no excuse. Apologies. I messed it up both times, and this is even our first attempt at recording. So hopefully you will forgive me and are excited to talk about things that are science, things that are sort of science, and things that wish they were science with us here. Jen, do you want to remind folks about what your lab focuses on and sort of set the stage for what we're about to discuss with your two former students?
Sure. We're basically all things yeasty. We use yeast as a model organism to address how different contaminants, stresses affect cellular metabolism. So we work on chemicals like Roundup, a coal cleaning chemical called 4-methylcyclohexane methanol, copper nanoparticles, and now we're starting to venture into starvation.
And so, Matt, I wanted to talk to you a little bit first, and Jen sent me two of the papers that you and her worked on together while you were her student. But before we talk about that work, can you tell us a little bit about where you are now and what you're doing currently?
Yeah, I'd be happy to. So right now I am the research and development scientist at a company called Imperial Yeast. And basically we grow yeast in big tanks and we sell it to breweries and anyone else that could use yeast. So that's where I'm at now. I've been doing this for a little bit under a year. I started this as soon as I graduated from Jen's lab.
And where is the Imperial Yeast Company based out of? Where are you in the world, I guess, is my question.
I'm in the Pacific Northwest, so I am in Portland, Oregon, and we actually opened up another location in Philadelphia, PA. Mostly here at the headquarters, but we're on the East Coast as well.
Cool. And so Jen sent me two of the papers that the two of you worked on together. One of the things we like to do on this show is sort of take as big a picture perspective as we can to make sure that everyone listening is up to date on the fundamentals of what you're doing. And so one of the papers that Jen sent me is from the journal Fermentation. And the article title is Saccharomyces arbicola and its hybrids' propensity for sake production. Interspecific hybrids reveal increased fermentation abilities and a mosaic metabolic profile.
And yeah.
A thing we like to do here on the show is ask people to explain the titles of their papers because there's a lot of words there. So I have some words highlighted, but if you want to take an initial stab at what the title of this paper is saying, I'll leave you to it and ask questions as they come up.
Yeah. Happy to. Glad I made some $5 words. Usually I don't get past the $2 mark. But so the title, it kind of changed as I wrote the paper. And this one, I tried to present the findings as clearly as possible. So basically, when we made the hybrids between the two yeast species, Saccharomyces cerevisiae, and the novel one, Saccharomyces arboricola, we made sake from it, and their fermentation profiles, so how much sugar they use, what kind of esters or flavor compounds they produced, we compared those, and what it revealed or showed to us was that basically the hybrids was a blending or a mosaic of the two parent species. So that's what we found. And I hope that kind of covers the highlights of the title there.
So when you talk about Saccharomyces hybrids, we have this so we on the show, we drink beer, we talk about beer a lot on the show. So hopefully our listeners are familiar with Saccharomyces cerevisiae, brewer's yeast. Cerevisiae being the Latin for beer, Saccharomyces being the Latin for sugar fungus. What is Saccharomyces arboricola? Where does this other yeast species come from?
Yeah, so it really dives into yeast nomenclature, and throughout history, that's been a pretty messy subject. Now, recently, it's kind of become a little bit more clear, but arboricola is a new species. And when I say new, I mean relatively new. I think like 2008, maybe it was discovered in China, and it's been, I think it's been isolated in Taiwan as well. I'd have to go back and look, but it's a separate species. And yeah, you mentioned Saccharomyces cerevisiae, and that's what we used to take a hard look at Jen's lab. But I think if you're talking about beer, there's basically two main varieties, if you will. And so you have an ale yeast, everyone drinks ales, and that's going to be your Saccharomyces cerevisiae. And then you have lagers, which are the Saccharomyces pastorianus, and that is going to be the hybrids between Saccharomyces cerevisiae and Saccharomyces eubayanus. So yeah, that's a hybrid. Saccharomyces pastorianus, it produces the world's largest volume of beer.
Oh, that's cool. I didn't realize that distinction. And I also know that, first of all, I want to say, Jen, jump in if I'm not asking the right questions and just be like, hey, here's the question you should ask. So Jen, please cut me off if I'm not going down the right paths here. But when we talk about Saccharomyces and we talk about these different hybrid strains, we often talk about sake brewing, but then we refer to sake as rice wine. And wine is typically fermented, in the Western sense of it, is fermented, not brewed. And so are you able to talk at all about why sake is considered a brewed product with brewer's yeast versus Western wines where it's a traditionally fermented product, or just clarify that distinction at all?
Yeah, so I think that it's called sake brewing, maybe just because translation can be a little rough sometimes. I mean, there's a lot of steps in sake brewing, but in Japan, sake actually means alcohol. So, it's kind of like there's many different styles of sake. So, in the Western world, it's kind of just diluted down to just mean rice wine, but there's a lot more behind that. And so, I guess as far as brewing goes, it's quite different. I guess they both come from grains, so maybe that might have something to do with it, whereas wine is.
That sounds about right, because for the rice, you have to boil it, and then you incubate it with this fungus that helps break up the sugars. Same thing with barley, is that you have to, malt it, and then you boil it to release the sugars. Whereas wine, you start directly with the juice. You don't have that step of releasing the starches into sugars.
Because the grape juice itself is heavily sugary, so you don't need to find a way to release.
Yeah, that's a good point.
That makes sense.
Yeah, it's the wine is, or I guess the juice would be fermentable, and then the rice and also the malted barley, those have to go through a mash process where it converts the carbohydrates to sugars. And so, the difference here is in how the enzymes do it. And beer, you're relying on temperature to kind of trigger the malt's enzymes to convert it from carbohydrates to sugar, whereas in sake, it's the Aspergillus oryzae or the koji that does the transformation.
Can you explain what you do? The Aspergillus. Sorry, I didn't even catch. I know koji. I've heard that koji term before, but can you explain what you just meant by those other terms?
Yeah, so Aspergillus oryzae, it's just a scientific name of koji. And I think if I remember correctly.
And so what is koji?
Koji is, it's Aspergillus oryzae, but what it is, it's a very similar to bread mold.
So we've gone in a circle.
Yeah, a very similar bread mold. I think Aspergillus is kind of, it's a filamentous fungi.
And what does filamentous mean?
Filamentous means that the yeast grow into the media or whatever they're growing into the yeast. So it's a polarized cell growth, whereas budding yeast, they just bud and they can't, sometimes they can, but in general, yeast don't grow in a direction. They just kind of bud off in a circle, whereas Aspergillus has this directional cell division, so it can penetrate into the yeast. Into the rice.
Oh, that's cool.
Yeah, if you ever see bread mold and you see how it's kind of stringy, those strings are the hyphae, and that's what digs into the rice. And so it actually digs into the rice and will put its nutrients or secrete the nutrients out of there via the hyphae.
That's really interesting. An additional question I had based on the paper that I read from Jen was that you were also interested in cryotolerant interspecific hybrids. And I'm aware enough to realize that cryotolerant just means cold tolerant, but I was curious about the fact that these cryotolerant hybrids have increased yields of ethanol and ethyl hexanoate. I don't know what the ethyl hexanoate is. And then you also talk about how that is an important flavor attributor to Japanese Ginjo sake rice wine. So I didn't know if you could just talk a little bit about both the cryotolerance, but also the secondary metabolites that you're trying to select for and what that means for the fundamental flavor of the product.
Yeah, so there's kind of two aspects here. And so the first one, the cryotolerance, which is cold tolerance. It's important because when you think about lager beers, most people understand that lager beers are fermented at a little colder temperatures and the yeast sinks to the bottom. And so that's relevant because Saccharomyces cerevisiae, it grows pretty warm. I think 30 degrees Celsius is optimal, and I think it grows it doubles every 90 minutes at those conditions. But when it's a lower temperature, these hybrids are able to grow faster and ferment, which, lagers are generally associated with, with the Germans. And I think it might have, there's some speculation, but it might have developed, when they had to storage their beer in the caves, and it was colder in the caves. So that's why it might be important. Also, when it's colder, you kind of preserve some of those finer volatile metabolites. They don't boil off or they don't evaporate. So I guess that's how that would be relevant. And it kind of connects the two because now we're talking about those volatile metabolites, which would just be flavor compounds. And ethyl hexanate would be if I remember right, it's the apple flavoring. And
cool.
The other one I talk about is isoamyl acetate, and that's going to be the banana kind of flavor. It's really, those two flavors are important in Ginjo sake, but they're also important in beer and brewing as well. So if you like beer and you like a Hefeweizen, they typically have some banana flavoring to them, and that's the isoamyl acetate.
Well, that's awesome, and I appreciate you clarifying that. I would like to talk briefly a little bit about the other paper Jen sent me, and the title of which is intimidating even to me: Metallomic and Lipidomic Analysis of S. cerevisiae Response to Cellulosic Copper Nanoparticles Uncovers Drivers of Toxicity. Wow.
I try
A
to
lot
make him
of
as
really
scary as I can.
interesting terminology there.
Yeah,
I'm terrified.
I might
Can
have
we
gone up
start
to
with
$6 on that one.
the metallomic lipidomic?
Yeah, so metallomic.
I'll pay whatever it takes to get you to explain it to me.
Okay, I'll meet you in the middle. So, yeah, the metallomic,
Appreciate it.
anytime. Metallomic and lipidomic. Let's start there. So the omics part is kind of an ending for anything that incorporates the whole picture of it. And so, metallomics just means the yeast cells entire metal profile. And so by that, I treated yeast with nanoparticles, and then I looked to see how the metal levels inside the cell change. So that's metallomic. And then lipidomic would be the same thing, looking at the entire lipid profile of a yeast. Once you treat it with the copper nanoparticles, we see how it changed. So that's the metallomic profile and the lipidomic profile.
Can I pause there?
Sure.
To just ask, like, what does that actually mean in the laboratory sense? Like, what are you doing when you walk into the room? When you say I did a metallomic treatment, what are you actually doing?
Sure.
What are you picking up? What are you putting down? What are you squirting into what? Like, just that whole situation.
So Matt, let's step back and start talking about what the copper nanoparticles are.
Yeah.
That too.
So I will talk about the copper nanoparticles. I will add too, but when you go into the lab and you're trying to do a metallomic analysis, what you don't want to do is burn down any part of a lab.
Oh, no, you're not bring that up.
I'm not going to tell any more than that, but that's a big no. You got to be careful because you use nitric acid and heat. So, but copper nanoparticles, yeah, what are those? So carboxymethylcellulose is the CMC part, and it's a cellulose derivative. And that really came from one of the people in the Wood Sciences division at West Virginia University. So I'm not going to speak a whole lot about the cellulose part of it, but it basically has a high ability to receive copper nanoparticles. And so it's kind of a hybrid material. It's part cellulose, but also part copper. And the copper part is, I think, about 20 nanometers in diameter, circles.
So these are antimicrobial materials that we were testing. So copper is a potent oxidizer.
Yes.
And so is it copper's oxidative status that makes it antimicrobial? I guess I've always been a little confused about like why it's not like we're dealing with a worldwide pandemic where this is important, but I've always been curious of like why copper and brass and other metals are considered antimicrobial in a way that gets promoted in the pop press and in advertisements. Like, what is it about these metals that makes them antimicrobial in a way that is relevant to the research that you and your lab are doing?
Yeah, it's really fascinating, actually, the whole aspect of copper killing microbes. One of the coolest stories that I found when I started looking at it was, I think the old hospitals used to use copper for their door handles and kick plates and stuff like that, where there's hot touch points. I don't think it's too expensive for most modern hospitals to do it now, but I think they used to also purify water that way. Isn't that right, Jen, in a copper pot?
It's part of Ayurvedic medicine where they'll put water in a copper bowl. And so the chemistry is that these metals will strip excess hydrogens. And so that oxidizes anything that it's touching. It's kind of like bleach.
It also depends on, what species of bacteria, yeast. I mean, in general, the bacteria is more sensitive to copper compared to humans. I mean, people wear copper bracelets, for instance, and it's not killing their cells.
And so what was this? What did your study find in terms of the yeast response to these various pressures that you were putting upon them in terms of the nanoparticles?
Yeah, so the drivers of toxicity part of the title, we looked at a couple of genetic factors to this. We believe that maybe the cellulose part is, or maybe the copper part, some part of that hybrid material is triggering some endocytosis mechanism.
And what does endocytosis mean?
It's cell eating. So basically it tries to import things from outside the cell into the cell. There's a couple different mechanisms behind it, but it's basically trying to take things in.
So the thing that we were puzzled at the beginning is we can measure the amount of copper that's in solution, and we can measure the amount of copper that's bound to these copper nanoparticles. And if you have the same amount of copper in each tube, you would expect them to be equally toxic. But the copper nanoparticles were 2.5 times more toxic for the amount of copper they had. So we were trying to figure out why were they more toxic, because we had this idea that the copper nanoparticles were sitting on the outside of the cell and oxidizing the cell membrane and the cell wall. But then Matt started following this line of investigation where we're like, maybe the cells are internalizing these copper nanoparticles and poisoning themselves from the inside out.
And that was something that stood out to me, was that this idea that no genotoxicity was found, and I would love to get a brief definition of what genotoxicity means, but then also the final line of the abstract I found almost haunting, was that like humanity must look forward past traditional measurements of toxicity. Like that feels portentous. It feels like you're saying something very profound and big about how we assess what is toxic within our environment. So yeah, the two questions fold into one big question, and I'll shut up now.
I think humanity needs to understand life more. I mean, that's the burning question we all try to answer, trying to understand stuff.
It sounds like you're doing a Matthew McConaughey impression. I don't think you're doing it on purpose, but like it's very effective in the sort of all right. We just got to understand toxicity better.
Yeah, call Lincoln. Lincoln needs to call me. I'm a lot cheaper. Yeah, I think we do need to understand it, and I think.
Can you tell me about genotoxicity? I would love to just for a definition of that term, because that term was unfamiliar to me.
Yeah, Jen's really good at defining terms, but to me, I think that genotoxicity, it doesn't have anything to do with, with the genes. That was a bad one. Rob, X that one out.
So like I was talking about how copper oxidizes thing. I'm cutting you off.
No, I said he threw me off with the Matthew McConaughey.
Okay.
Dude, you are so... Dude, if I'm the only person that tells you were doing a fantastic Matthew McConaughey impression, our listeners will be a disservice, because you nailed it. You were so good.
Too bad they don't see the video of him leaning back in his chair.
It was so good. It's absolutely a compliment. It's not meant as a criticism at all. But like, you just were so smooth and charming in your explanation of the yeast toxicities.
Okay, so we can get back to genotoxicity. We were talking about how copper nanoparticles damage biological materials by oxidizing them. And one way to kill a cell is that you basically break all the DNA, you've oxidized it. And so in the field, that was the primary way we thought that the copper was killing the cells, by basically breaking the DNA and damaging the genome, which is the genome toxicity. But what Matt found was that the primary damage was at the lipid level, which makes sense if the copper nanoparticles are sitting on the membranes. They are poking holes in the membrane by oxidizing these lipids.
That's right. We did the pulse field gel analysis on that.
Yeah, the pulse field gel measures double-strand DNA breaks.
Yeah, and I'd like to go into a little... I was going to say, I'd like to just kind of bounce off what Jen said earlier about the nanoparticles being different than regular soluble copper. It seems to be a real trend in nanotoxicity or nanotechnology in general. And so, quantum nanodots is kind of central theme. And in quantum nanodots, I might get some of the details a little bit wonky here, but I believe it's silica or some other, but it's all the same material. And based on the size alone, the color changes. And so it's kind of like once you reach this nano scale aspect, things kind of play by a different set of rules. It's like, one of the most striking things is gold. Everyone knows gold is yellow. But once you get down to certain nanoscale diameters, it changes to red and shades to red. And that's actually, if you go to a church and you have a stained glass window, and it's from, olden times, chances are that red is made of gold nanoparticles. They didn't understand how to do it. I mean, they understood how to do it, but they didn't know what they were doing. They know they got red.
And I guess the sort of question that I want to round up this segment on is, Matt, you answer this however you want, but like, what's the big takeaway from the work you did during your thesis research or what you're doing now? Like if there was sort of one lesson you wanted people to learn from what you do, what would that be?
Yeah, that's a really good question. And this is my second podcast I've ever done. So the first guy asked me that same question at the end, and I totally fumbled.
What?
I just started blathering out. I did. It was the Brulosophy guy. He's starting a new thing out. But no, it's a great question. And if I had to pick something, I would say for the copper nanoparticles, toxicity really boils down to two things. It's not necessarily just the element copper. It's what form of copper and how much. I mean, it sounds very simple to say it, but it really depends on, poison is in the dose, and it really depends on what type and form of copper. So that's the summary of copper nanoparticles of 5 years of research. And then if you go to the hybrids, the hybrids, I think you can relate interspecific hybrids in yeast the same as a cross between a horse and a donkey. You get a mule. And so, this is very common in livestock. It's very common in agriculture. And it's less common in microbes. But I think for food production, specifically for beer, I think that by crossing Saccharomyces cerevisiae or Saccharomyces species in general, what we can get is we can get an increased diversity of flavor, but we don't have to risk the spontaneous fermentations, because with Saccharomyces cerevisiae and inoculating or pitching, as the brewers call yeast in there, we get a reliable fermentation. So we're looking to find maybe a new type of lager yeast. So, just trying to mix horses and donkeys.
You're just trying to find a new mule. I think that's a beautiful answer.
Just trying to find
I love it.
The mule. Yep.
And
not a Moscow mule.
I think a Moscow mule would be a fantastic thing to be drinking if one were to be drinking, which one could then promote on our next segment, the What Are You Drinking segment, coming up next here on Science Sort of. An episode of Science Sort of we didn't also talk about what we are drinking. Apoorva, you have been super quiet because I've been talking to Matt for a little while. So since I suspect you have the most morning focused beverage, would you like to tell us what you are drinking?
Yeah, I'm not keeping up with the yeast theme of today, but I'm drinking a hot cup of coffee right now, and it's pretty good.
What kind of coffee?
It's just a dark roast, French press, simple cup of coffee with cream and sugar.
Are you normally a dark roast person? Is that your typical preferred roast profile?
Yes, and we had the most fabulous coffee place in Morgantown, which I really miss. But yeah, always been a dark roast person.
What was the place in Morgantown?
The Grind. I'm sure Matt and Jen
Woo!
could agree. Yeah.
It's the closest coffee shop to the lab.
Oh yeah, and sometimes you just need coffee to make the day better.
I like to go there and work and look out the window, watch people.
And rescue the hand.
Real old man
Remember
style.
you saved that fainting woman?
Oh yeah, some woman passed out and I had to catch her. She went whoop.
Wait, you got to tell the whole story there, Matt. It's not a full story. What you just said. You just said whoop,
that's pretty
and
much
then
the whole
made
story.
yourself a hero.
I don't know about hero, but I was definitely the closest person to her. There's like 5 seats looking out the window at this coffee shop, and the lady beside me was watching a YouTube video or doing an assignment or something, and she kind of just went limp, and I think she started to lean towards the other person beside her, and I noticed, so I kind of just caught her before she hit the ground.
Oh wow.
And then I had just taken a Wooford course, which is the wilderness first responder, so we just checked her out, and she came back to it and said she had done it a few times before and just waited for her husband to call, or she called her husband and waited for him to come, and we just kept an eye on her. So I really didn't do much except for, not let her fall.
Wow.
Well, given that heroic story, I have to ask, what are you drinking now that you're a hero?
Well, I wish I was back at the grind watching people walk by the street. What I'm drinking now is a Suss It Out Rye India Pale Ale from Level Crossing. It's one of the perks of working at a company that produces yeast for breweries. You can go into pretty much any fridge and have your pick. But yeah, that's what I picked up, a little rye IPA.
And how does it taste?
Well, it's pretty hoppy, and looks like, according to the back, the color is on the lighter side. It's in a can, so I can't really see it. But it's pretty good. It's pretty good. Pretty good. What about you, Jen? Hope I'm not stealing your thunder. What are you drinking?
If you can see, I've got baby cakes because you guys are my scientific babies, and you've grown up. So this is from the Tribus Beer Brewery that's in Milford, Connecticut. I went there a couple weeks ago. And it's a stout.
Nice.
It's very coffee stout. It's good. It's a little lighter than I'm usually drinking. It's 5.5%, so good for the evening.
Okay. So Apoorva's got the coffee, I've got the beer, and Jen's got the hybrid between the two.
Right?
Uh-huh. Now I think we're catching the dynamics of the old lab crew.
What was the dynamic? Did you guys, what was the lab,
Apoorva was a rock star. That's the dynamic of it.
Everyone got along, but everyone hated Matt. So other than that, we were great.
Well, no, it was, if you couldn't find it, did you look on Matt's bench?
I put it back sometimes.
Sometimes you put it back. Yeah. Well, they were baymates, so they were back to back. And then I still have my bench on the other side, although it's been a long time since I've physically been at the bench.
Yeah, I guess if there was anything weird, you could look in Jen's desk. She had a drawer that you kept weird stuff in.
It was the drawer of all the things that you didn't know what they were, so I just stuck them in my drawer. What do you have tonight, Ryan?
I am having okay. Background. This is not the first time we've tried to record this episode. So I had a different beer lined up for our previous attempt. I think the beer I had previous to this beer was better, but I'm still happy with the beer that I have. So I'm having a Weathered Ground Brewery Hop Farmin Miner. It's a West Virginia brewery.
Have you been there?
I've not been there. It's in Coolidge, West Virginia. I've driven past it, but I haven't had a chance to stop there because there's this thing called COVID-19. It's a bit of an issue. We've been dealing with it for a little while. I think we're gonna be past it in a week or so. So
Yeah, we just have to lean into it hard for 2 weeks.
It's so depressing. But I just wanted, I know you all are not necessarily native West Virginians, nor am I, frankly. I moved to West Virginia and have adopted it as my home state. But I just wanted to I wanted to feature something from West Virginia with this crew of folks who have all spent a significant amount of time in West Virginia. And this is a beer I've been saving in my fridge for a little while. I bought it at Tip Top, the coffee shop in Thomas, West Virginia. Folks are familiar with the beautiful little town of Thomas and the great coffee shop. Honestly, one of my top coffee shops in the world, Tip Top Coffee in Thomas, West Virginia. They also have mixed six packs, and I bought this one beer as part of a mixed six and have been enjoyed all the other beers and then saved this beer because I was like, this beer is going to come up on a show at some point. I know it. And it's in a can. So I can risk the wait. And also, I was preparing for this episode, and all of you do Saccharomyces cerevisiae work, so any beer I have is on theme because it's all brewer's yeast.
Although I have to make a correction. I have to make a correction. Matt is a native.
Please do.
He's a native West Virginian.
Yeah. I was born and raised in Parkersburg, West Virginia.
I mean, Parkersburg, you're practically from Ohio.
Marietta's across the bridge.
It's okay. I lived in Ohio too. My dad's from Ohio. I got a lot of family from Ohio. It's not necessarily an insult.
No. I got a couple of jokes about it, but I'm going to keep that for another time. Jen, isn't coffee? Coffee's fermented too, isn't it?
But is it fermented by
the beans are fermented?
Beans are.
I think it's mixed. Right? There's got to be mixed micro.
Yeah.
Is it
just
really
done in the field. I think so. I might be wrong.
Didn't think there was any yeast in there. I have no idea.
I think it's definitely oxidized. I don't know that it's fermented.
I don't know. You guys want to put a wager on it? Just want to make it fun. I bet a drink.
We can put a wager on it. We can put a wager on it for the episode where y'all come back to talk about future updates to research. But we have been remiss because we have yet to talk in depth about Apoorva's research. And that's what is coming down the pike in our next segment coming up right now. I'm super excited to talk to you about the work you've done with Jen and otherwise. I'm looking at one of the papers that Jen sent me, and the title is Mitochondrial Metabolism is Central for Response and Resistance of Saccharomyces cerevisiae to Exposure to a Glyphosate-based Herbicide. I need a little help because I don't know what a glyphosate-based herbicide is or means or matters. So please help us.
So it's very interesting that you actually asked me that because like you, I think most of the people wouldn't have really thought about glyphosate-based herbicides and what these really are. But have you heard of Roundup?
Yes, I have.
Yeah, so when you say Roundup, people are like, oh, I know that. I spray that in my backyard all the time. So that's basically what it is. So there are tons and tons of glyphosate-based herbicides out there, and Roundup is just one of the examples. You have your glyphosate, which is the active ingredient, and they add different additives to this, and hence we get all the different commercial formulations that we have out there.
But what is a glyphosate, I guess, is one of the questions.
So glyphosate is basically this chemical component that is targeting this pathway that's called the shikimate pathway, which is basically the aromatic amino acid pathway. So if the cell needs to produce aromatic amino acids, which is important for plants and other organisms, it basically needs this entire pathway. So what glyphosate does is it inhibits one of the enzymes that acts in this pathway, and that's how it kills these plants. And that's how this entire thing works as an herbicide.
Okay, so we have this thing that's designed to kill plants, but you're interested in what it does to a fungus. And I think we haven't really touched on is Saccharomyces cerevisiae is a fungus. Can any of you, Apoorva, you're right at first refusal, but when we talk about Saccharomyces as a yeast, as a fungus, what do we mean by that? Like, what does it actually mean that this organism is fungal relative to it being a plant or an animal? And just, can we talk a little bit briefly about that distinction?
I would say yeast falls in the kingdom of fungi just based on its genome, how it's placed. But it is a pseudofungus because it doesn't form the hyphae that Jen was explaining earlier, just based on its structure and how it buds out. That's a unique quality, which is why they call it a pseudofungus.
Okay, that's always a question I've had. That's super interesting. Okay, can any of you elaborate a little bit more on the hyphae situation?
Does Jen want to jump in? It looked like you had something to say.
Well, I wanted to say that.
Does.
So fungus that we think of, mushrooms and stuff, we think of them primarily as digesting decaying matter. Whereas plants, which also grow out of the ground, they have chlorophyll. So they are green and make their food by using sunlight. So that's the primary way we differentiate by eye if it's a fungus or a plant, is that fungi don't have chlorophyll. They need to acquire their sugar from whatever they're growing on, and typically that's decaying matter. The fungus, Cerevisiae, is a single-celled organism. So it doesn't form a mushroom, but it's related to those types of organisms. And plants, fungi, and humans, we all have our DNA inside a nucleus. Bacteria don't have a nucleus. They also have their DNA circular. So plants, fungi, and humans all have their DNA. It looks like a little X, their chromosomes. Yep. Whereas bacteria just have a circle. And so bacteria are going to be sensitive to antibiotics. Fungi are not sensitive to antibiotics because they're not a bacteria. And the shikimate pathway that we're looking at exists in plants. It also exists in fungi. So they can make a subset of these amino acids that we call aromatic amino acids. Humans don't have this pathway. That's why Roundup and these glyphosate-based herbicides are not acutely toxic to people, because we don't have the pathway for glyphosate to inhibit.
So what you're saying is if we were exposed to a glyphosate-based herbicide, would experience no ill effects, but fungi, especially these Saccharomyces fungi, do experience a negative effect.
Technically, yes. Yeah, that's basically what my research was on, because, logically, if we don't need that pathway, so glyphosate shouldn't affect us at all because we're not making the aromatic amino acids. But what my research basically talks about is how all these other substances that they're adding, the additives that are in there, which are supposed to be inactive or have no effect, when you put them all together, we're not testing them enough. So sometimes they do have adverse effects. That is where we're testing the cell to see what else is getting affected when you treat it with these commercial formulations as it is.
So I actually think that transitions us nicely to the second paper that Jen sent me, the paper that was published in Genomics, G3, Genes, Genomes, Genetics, which is called Resistance Mechanisms of Saccharomyces cerevisiae to Commercial Formulations of Glyphosate Involve DNA Damage Repair, the Cell Cycle, and the Cell Wall Structure. And in that paper, you explicitly say, though labeled inert, when used individually, certain commercial formulations of glyphosate have adverse effects when used in combination with other additives along with the active ingredients. So can you talk a little bit about what you meant in that sentence?
Sure. So this sort of brings me back to like a question you were asking earlier about this is basically about plants. Why study this in yeast? How does any of this matter? So in yeast, we just think of it as a species, but within the species, there is so much diversity. And we basically use that diversity within the species to try and understand what is getting affected by these different things that are present in these commercial formulations and how it's getting affected. So when I say there's diversity, there's so much variation within it, such that some yeast strains are sensitive, some are resistant to these chemicals. So we try and figure out which ones are sensitive, which ones are resistant, and basically what is the difference between them, which tells us what's making it sensitive or resistant. It's all these other chemicals that are in there are tested individually, correct? To make sure that it's safe for us. And when we're exposed to it, it's not really a problem. But they don't do enough testing with a commercial formulation as a whole, because with glyphosate-based herbicides, a common problem you see is things become resistant. You're using it in your yard, and a couple of months later, you're like, it's not working anymore. Maybe I need a different formulation. So that's just tweaked a little. They've changed a few inert ingredients in there, and that makes it sensitive again. But that should tell us that when you're changing these combinations, something in there is changing, the way it behaves, the way it interacts. So unless it's tested enough, we don't know if it's safe or not, or what these adverse effects are when they all act together.
So if I was to sort of take the analogy you used in the paper and bring it forward into the discussion we're having here, it's sort of like we have a type of yeast that may or may not be susceptible to compound A, and so we test it against compound A, but then we also are not sure if it's susceptible to compound Q, and so we test it against compound Q, and it's fine against both A and Q, but the thing we're missing is what happens if it's exposed to A and Q simultaneously. And so we're missing that sort of combined exposure to the two things that it can survive alone, but can it survive them together?
Exactly. It's sort of like bringing it back to what Matt was talking about earlier in terms of how there are processes where you need the fungi to actually break down the carbohydrates and give you sugar, and then the yeast can act on the sugar. So if you put them in individually, it may not give you the final product that you're looking for. But if they're both in there, you get your beer that you're looking for or whatever, because there are interactions and other compounds that are coming into play. So the same thing goes for this as well.
So I guess what is the thing that you're hoping to find on the other end of the exposure of the yeast to the compounds that you're exposing them to? Like, so what's sort of the outcome that you're trying to measure?
So the idea behind this was to try and understand at a cellular level or at an omics level that's looking at the entire thing, what are all the changes that are happening within the genome, to look at every single pathway, every single gene, to see what are the things that are changing, because then we can see, are these genes are they belonging to pathways that are actually conserved? So does it actually affect us because those other pathways are present in us as well, though we don't have the aromatic amino acid pathway? So the more we understand about what are the genes and pathways affected, the better we can interpret, are these formulations actually safe for exposure or not?
And so what did you find?
We found that there are a lot of pathways that are actually affected. Mainly, we were talking about how there is some amount of mitochondrial DNA that's affected, cell walls that are affected, though we don't have cell walls as human cells. But yeah, mitochondria.
Well, wait, you say that like that's like a super obvious thing. But I think that's my job, is to be the idiot in the room and to be like, hang on, why is what you just said a fundamentally important part of the science you're doing? So you said humans don't have cell walls, and I believe that applies to all animals, right? But fungi and plants do have cell walls. So can you just clarify that a little bit, just so everyone's on the same page?
Yeah, so plant cells and fungal cells, they all have cell walls, whereas any animal cell doesn't. So that part of it is not really applicable to us as humans if there are any variations in cell walls. But a lot of other pathways are getting affected, such as like whether you're talking about mitochondrial DNA or you're talking about any cell cycle proteins, DNA damage proteins. These are all present in humans as well. So it's important for us to know if there are any changes brought about in these, because it could potentially affect us over a really long period of time.
That's so fascinating. I love that. So I'll ask you the same question I asked Matt. Sort of, what is the sort of big takeaway lesson that you would like people to know from the work that you've done over the past couple of years, in Jen's lab and beyond?
The big takeaway for me definitely was in terms of the usage of certain terms, in terms of like when they study glyphosate-based herbicides, they're either just studying the active ingredient or they're studying like one particular formulation like Roundup. But everyone just calls it Roundup, even if it's Roundup Pro or different formulations. So use the right terminology when you publish so people are actually trying to understand what all these other inactive ingredients are doing in there, as well as we need to like thoroughly understand when all these interact, how they're affecting the cell at a cellular level, what's changing in the genome. So basically a lot of omics studies.
And just, I know you mentioned the term before, but just so we can be fundamentally clear, can you tell us what you mean when you say omics studies?
I mean, in terms of looking at the entire cell as like the genome, the proteome, transcriptome, to simplify that, just in terms of all the genetic material that's in the cell, all the proteins that are in a cell, looking at it as a whole and not just focusing on one particular target gene or one pathway. Would you define it like that, Jen? I don't know if...
Yeah, I mean, for omics studies, we use these as large measurements. So you're not specifically measuring one thing. We're just measuring everything.
Yep.
And then we use statistics to determine which components are the ones that are changing in levels.
Yeah, I think I'd have to echo what they said and probably add on. It can be targeted, but it's less targeted and more holistic view of the cell. Like, how is it changing the cell in general, but with mega data sets.
Right. So it's a play on the word of genomics. So we got genomics.
Right.
Proteomics is protein. Transcriptomics is the transcripts, the gene expression. Lipidomics, all the lipids. Metalomics, all the metals.
All the omics.
I mean, I decided early in my scientific career that like I wasn't going to study anything smaller than what I could tackle, like physically just like bring to the ground. And then I ended up doing stable isotopes, right? Like isotopes being one of the smallest fundamental particles of the universe. So like I botched my own plan, but my fundamental goal was always to focus on things bigger than the microscopic scale, just because I recognized that my brain wasn't quite there for the sort of cellular level analyses required to do the sort of work y'all are doing. And I think it's amazing. I have a hard time grasping it, I guess.
Well, it's like learning a different language. You just have to become fluent, and you just get intimidated when you are outside the field because of all the words.
But it's only 4 letters. How could it be that hard? A, C, T, G, right? Those are the 4 letters I have to learn. But I should be able to speak the entire language. Just turns out it's still a little tricky.
I'm still learning.
Oh, yeah.
Apoorva, can you tell us a little bit about where you're at in your current research and where you hope to go in the future, and sort of like what you're hoping to learn, and sort of if folks are interested in learning more, where they can touch base with you and follow along in what you're working on?
Sure. So when I came to Jen's lab, that was the first time working with yeast for me, but I guess I found it so fascinating. I was hooked. So I'm currently a postdoctoral fellow, and I'm still working in yeast genomics. And as Jen was saying, I'm still learning. There's just so much to learn, and it brings you to like new avenues, and you always find different things that you haven't done before. And I think that's been the best part, just getting to learn new stuff and do new stuff every day. yeah, though things have changed in terms of what I study now, I still study yeast, and the diversity of yeast plays a big role in my research. If anyone wants to reach out, shoot me an email. It would be awesome to talk to people, answer questions, and talk more about research. I feel like that's something we all have in common. You can't get us to shut up once we start talking about our research because we think it's really cool. So, yeah.
A thing I often point out to folks who are not scientists is there's this idea that like, oh, scientists don't want to hear from me. You know, they don't want my email. And I'm like, if I ever got an email that was like, I read your paper or I listened to you on this podcast and I have this question about what you said, I would be thrilled. I would be so thrilled to get that email. Like, there's no other way to say it. It'd be so great to just get a person who actually cared about what we were working on.
Exactly. And the other part of it, I think, is also that we try to like scientific communication is such a big part that you try to simplify and use terms so people can actually understand what you're doing. And you feel like, oh, there's one person out there who actually understood what I said, and that makes it totally worth it.
No, but that one email can make your month.
Definitely.
Right. Matt, same question.
Well, I think I work for a yeast company, and I'm in the research development lab. I'm actually starting up. I'm the only research and development guy. So, I'm continuing on brewing science research. We've got some projects coming down the pipeline. So, and that's the exciting stuff for me is, I'm looking at how to do things with yeast that haven't been done before. So it's less fundamental. Apoorva's research is fundamental, and I don't know where she's going with it. And Jen's always been really fundamental, but I've always kind of geared more towards application based. So that's kind of where my head and I guess my heart is at. And I don't know exactly where it's going. I got some projects on the board. So that's kind of where I'm. That's kind of where I'm standing. I would like to. There was one thing that Apoorva said that kind of brought a bullet point back to my head, and she was talking about the diversity in yeast and how it's pretty incredible. And so, in brewing yeast, at least, there's an incredible diversity of phenotypes. And I think it was a Gianni Liddy paper that I was reading, but it explained the diversity between Saccharomyces cerevisiae strains and not species as the genomic difference between man and chimp, and then between Saccharomyces species, the diversity there is the same genomic-wise as humans to birds. So there's quite a large level of diversity within Saccharomyces and Saccharomyces cerevisiae. That's a little tidbit I always like talking about.
That's fascinating. I did not know that degree of genomic diversity existed within that sort of, the whole idea of the phylum, order, class, genus, species level existed. Before I let you both go, though, Matt, I'll ask you first. What would you like to say to folks who might be interested in pursuing a career in yeast science? What would be your lesson that you would want them to learn as they embark on that career, and advice that you might have?
Sure. Yeah, so Saccharomyces cerevisiae. It's the fungus among us. It's the coolest, I think. but on a serious note, I started learning about Saccharomyces cerevisiae first in brewing terms in like Bio 101. And when I was a senior in college and undergrad, I started learning about its real applications for research, and that a lot of the genomic and molecular biology was happening there. And I was at Fairmont State, which is a smaller school, D2, if you play sports. And my professor was like, hey, I asked him, I was like, hey, can we do any of this type of research? And he said, well, you can't do that here. You have to go to a major R01 institution like West Virginia University. So that's what I ended up doing somehow. But as far as advice going, if people want to research yeast, I think reaching out to whoever is at your local university. That's what I did. I found Jen that way, and I know that I would be happy to answer and field any questions. And I'm sure my colleagues here, Dr. Gallagher and Dr. Ravishankar, would be happy to talk to anyone.
That's awesome. Apoorva, what about you? What advice would you, if we do have young people who listen to this show, what sort of message or lesson would you want to impart upon them as they pursue whatever STEM career they might be capable of accessing?
If we're talking about yeast research specifically, for me, I thought it was cool because if you want to take the route that Matt's taken and be more applied, you have those opportunities, whether you're working in brewing or various other industries, or what floats your boat is more things like fundamental research, which is what I really like. You can go that route as well. But basically, I'd say research is something that's just exciting for me. I like asking interesting questions, and irrespective of what the question is, figuring out those answers, coming into lab and being able to see results. If that's something that excites you every day, definitely go for it. Like, it reminds me of what Jen used to tell me when I initially started off in her lab. She used to always say you could teach people so many different things, but you can't teach them to be enthusiastic and be excited about your research. So if you feel like you're excited about research, this is definitely something you should pursue.
Well, it sounds like all of us owe Jen a little round of applause for producing such wonderful students and being just an awesome person and professor generally. Matt, Apoorva, thank you so much for joining us. I'm going to hang on to Jen for a little bit, but before I let the both of you go, is there anything, any links, any resources, any social media stuff that you would like listeners to our show to be aware of as they hear you and get and frankly get excited about the work you're doing?
For the links and sources, I will leave that up to Matt because I know that's completely his forte, and I'm sure he'll have advice for you on that front. But I just wanted to say thank you to you, Ryan, for having us on the show. It's been so much fun to talk about research in general, as well as everything we did in grad school. And thanks to Jen for being a phenomenal mentor.
Thanks.
Thanks, Ryan, for having us.
Of course.
As far as the self-promotion, I'll throw a little plug in. Yeah, I have a website, mattsyeastlab.com or .org. And then you can find me on LinkedIn or, if you guys or anyone out there is a brewery wants to order yeast from us, I work at Imperial Organic Yeast. We're on the web. It keeps me in the lab. So that's my plug. I'd like to echo again, thanks, Jen. We already talked today earlier, so she knows how much I appreciate her. So I'll just leave it at that. And thanks, Ryan, for letting me stay corny with the fungus among us thing. I was waiting the whole time to say
that. No, my pleasure, man. It's we are a pun-filled program for sure. You both were fantastic. Jen's been on our show before. Jen's been a listener to our show, so she knows the nonsense that we're all about. But I believe Jen is the first person to bring her own grad students on the show and make them face the gauntlet that is my nonsense. And so I really appreciate Jen putting her own students through that. No one else has had the courage, the gumption, the audacity to make that happen. I think it's great. I love talking to both of you. Thank you. This is an absolute delight. And I hope it's not the last time that you both appear here on our program.
For sure.
I really enjoyed it, and it sounds like Ryan's challenging some of his listeners to come on the show with some of their grad students. And if I'm going to put another pun out there, I would say he's a pretty fun guy.
Oh, my.
Mm-hmm.
That's all. I'm done. Now I'm just looking to see if there's a way I can hang up on Matt specifically.
It's the red button.
There's a way to just that looks like there's not a way to just hang up on Matt as much as I would like to. But
the fun guy was a compliment. Come on.
I know, but it'd be funnier if I could just hang up on you in the moment. So I'm gonna. Thank you so much to Matt and Apoorva for giving us so much of their time. Jen, thank you so much for bringing us some of your students. That was amazing.
It's hard to watch them grow up.
I think this might be a first for us in having an advisor sort of put her students through the gauntlet of being interviewed by me. How'd they do? How you feeling?
It was good. I mean, it's always hard to jump in. I have a bad habit of jumping in when I think I can help the explanations. But for the most part, I wasn't sitting on my hands the whole time.
Well, that's great. I loved talking to them. I thought it was great. If they decide to listen to the show in the future, they might decide to then themselves respond to the content that we presented, in which case they might end up as participants in our final segment where we deal with feedback from the listeners, and we lovingly call that the PaleoPOW. Coming up next. The PaleoPOWs is a segment of the show where we deal with feedback from the listeners. We have the best listeners. They give us the best feedback. And so it's so much fun to get to talk about what that feedback means, what that feedback is. I want to start by featuring a recurring PayPal donor from George H. We don't allow people to sign up for recurring PayPal donations at this point. If you want to become a recurring donor, you will need to sign up for it via the Patreon link. That is patreon.com/sciencesortof. Patreon.com, you get a ton of great rewards when you sign up, and we really, really appreciate it. It is what enables us to keep the show going. We love that George H gets to keep supporting us via PayPal. But if you are not George H and have not yet subscribed via PayPal, we hope that you will consider supporting us via Patreon. And we thank all of our Patreons who do. I have one other bit of feedback. This is an interesting bit of feedback. It comes from Jen G. This is based on Mammoth Monday, our episode where we talked with Advait and Lindsay about mammoths and mastodons. And the comment I'm going to read, and then Jen, I'll let you respond to, if that works.
Yeah, that works.
I think it'll be interesting. So Jen G wrote in, and I'll just go ahead and say the G is for Gallagher.
There's no secrets here.
Hey, Ryan and the Science Sort of crew, congrats on defending your thesis. That's my thesis, not the whole crew's thesis, but we don't have to get into that unless Jen insists, in which case I'll be embarrassed. In the Mammoth Monday episode, you mentioned sequencing mitochondrial DNA instead of nuclear DNA. I can add some more info on why. Mammoth, like humans, have two sets of nuclear chromosomes, one from each parent. The mitochondrial DNA, for the most part, is only inherited from the mother and depends on which tissue. There can be hundreds of to over 1,000 mitochondrial genomes. Tissue like muscle, which had more energy demand, will have more mitochondrial DNA because it has more mitochondria, which is why it's much easier to detect mitochondrial DNA, even though it's smaller than nuclear DNA. There are so many more copies per cell that the PCR reaction used to detect and amplify it for sequencing is easier than nuclear DNA. Happy holidays and good riddance to 2020. Bye, Jen Gallagher. So Jen, you sent me this comment. This comment was sort of what spurred our discussion here that has occurred today in this episode. And so I just wanted to give you an opportunity to sort of comment, clarify. I mean, you're absolutely right about the mitochondrial DNA versus the cellular DNA. So I was super excited to have you on this episode to talk about what it is that comment meant and what you were saying in the moment.
Yeah, I mean, for a lot of the science that you talk about, I'm the dummy in the room. And so I've learned a lot from that. But in this case, I was screaming at the phone going, because there's so many more copies, because I think it was you or somebody else was a little puzzled why
Let's
the
say it was
mitochondrial
me.
DNA. Okay.
Let's say I was the idiot.
We can all play the idiot sometimes. Yeah, so the mitochondrial DNA, the mitochondria are these small organelles that produce the energy for the cells, and they are primarily inherited through the
I
egg.
hear they're the powerhouse of the cell.
They are the powerhouse, and they will take the ethanol that we have been drinking and convert them into ATP and carbon dioxide. So energy intensive.
Beautiful. Love them for it.
Energy intensive tissue will have a lot of mitochondria, like muscle. And particularly when you're looking at samples that are very old, like those mastodons, the nuclear DNA tends to break up over time, as does the mitochondrial DNA. But because there's so many more copies, you will get some that's intact enough for the PCR reaction to amplify.
And so I loved that you wrote in and gave us the feedback that you did, because I will admit that I was a bit of a neophyte in that I did not fully understand that the density of mitochondria differed based on the type of cell. So like I joked, mitochondria is the powerhouse of the cell. But like I was guilty of that being sort of the be all end all of what I knew about mitochondria. I didn't realize that like mitochondrial density differed based on the type of cell that was being discussed. So am I understanding the distinction there, that like certain types of cell have different levels of mitochondria and that affects our ability to detect the DNA in them?
In the mitochondrial DNA, yes. I mean, I will make a plug for my lovely yeast. Yeast also can change the number of mitochondria they have depending on how they're growing. So if you're growing them in really rich media with lots of yummy sugars, there'll be fewer mitochondria, ironically, because they are, instead of taking those sugars, they're taking those sugars and metabolizing them to alcohol, which is fermentation. But what mitochondria do is they do respiration. So they do the next step.
Mm-hmm.
Fermentation generates some energy, but respiration makes a lot more energy. It takes more time. It also takes oxygen, which is why yeasts can do the fermentation in bottles without any extra air in there. Actually, you don't want air in there, because then you'll end up with the cells doing respiration and they'll eat that alcohol and turn it into carbon dioxide, water, and ATP. So they're really dynamic. If you look at mitochondria under the microscope, depending on how the cell is growing, they can be little tiny fragments, or they can be this huge lattice-like cage inside that looks like art. It's just really pretty.
I think the only comment I have there is you said ATP, and I just have to point out that I knew that meant adenosine triphosphate, right?
Uh-huh.
Can we reverse back to what exactly we weren't precise enough about when I came to our mammoth episode? And so if you can you might not remember, but the idea was that if we're going to clone mammoths using mitochondrial DNA, your point seemed mostly to be about like why that was from the mother and why the energy demand. I think you talked about this already a little bit. So like I think you've covered some of this, but like why the energy demand is based on the cell that was sampled. But I think the main thing is,
yeah, so mitochondria, there's always exceptions, but primarily is inherited from the mother, because the egg is where the mitochondria is. The sperm is so small that it doesn't, those mitochondria that power the sperm don't get in when they're fertilizing. So it's maternally inherited. Same thing with humans. You can sequence your maternal mitochondrial DNA and you will see your mother, you'll see your maternal grandmother, and it'll go down that line. And as I remember from the episode, they were looking at how many individuals and how genetically related those individuals were by looking at the mitochondrial DNA. So that will tell you maternal lineage. And because over time, there are small differences that will pop up, just like a coronavirus might change and make new variants just by random mutations. mitochondrial DNA will start accumulating random mutations. So you can look at that and kind of get a molecular clock to figure out, I have DNA from this individual. It looks like DNA from this other individual. And based on the small genetic differences, it's probably separated by X amount of time.
I love that explanation. That's awesome. Thank you for clarifying your own comment. If it wasn't clear to folks, Jen was the one who wrote the comment that I was referring to, and I was kind of quizzing her on what that comment meant. And I think, you've explained it beautifully. How's everything else? Everything going good? How's science life in?
Science life is a little slow at the end. So it just so happened that I had a lot of papers I had backed up. And so during the shutdown, I was working on getting the papers out. And then, part of the academic life is the hamster wheel of getting grants. So I have submitted 4 grants in the meantime, and heading back to the lab soon to start helping some of the students there begin new types of experiments. But I'm really hoping that in the next few months we can get back into the lab, because I start seeing the enthusiasm, momentum in the students starting to lag. But I've been at home. I have a little lab set up. I have a little Instapot that acts as my incubator. I brought my dissection microscope home. So that's been a little happy place.
This was an amazing episode. I loved talking to Matt and Apoorva, and I hope that it was not their last appearance on the show. But regardless of whether or not they come back on the show, that when we come back next time, we'll be talking a whole lot more science. Sort of. Audio production for Science Sort of is done by Rob Heath at Rob Heath Studios.
Visit sciencesortof.com for show notes, links to all the stories we talked about, and ways to interact with the hosts, guests, and other listeners. Science Sort of is brought to you by the Brachiolobe Media Network of podcasts. That's all for this week. See you next time on Science Sort of.
So my dog has learned terrible habits to whine at me whenever I'm recording to get treats. So Julie is going to form a support role here to prevent the absolute incessant whining of a spoiled puppy dog.